Okay, so then let's just dive right into the lecture. So this will be a very short recap of representation learning in general, because this will be the foundation. So this will be the input that we will then use later on to predict secondary structure, or yes, to predict secondary structure using CNNs, for example. But first of all, how can we actually represent our data, and what does this actually mean, represent our data? A representation in general converts an observation in the real world, and this can be anything from an image, a movie, speech, a word in a sentence, or in our case, a residue in a protein, into some mathematical form. For example, a vector that can be read by a neural network, for example, or by any machine. I mean, this does not even have to be machine learning related. This can be any computer. Because I mean, here you just see a very simple 3D sketch of the hepatitis C virus protease helicase. This is something we can nicely visualize for ourselves in 3D. We can color certain domains, subregions, functional subregions of this protease. But in order to make this understandable also to the computer, and especially to machine learning devices that would need to operate on these 3D coordinates, because essentially these are 3D coordinates. So you have X, Y, Z coordinates, and you would need to find some representation that the neural network then can operate on. So this is the overall goal of representation learning, to find novel ways to actually represent your data and make them as readily machine readable as possible. But just for the historic context, if you want to put it so, one of the first and most straightforward options to actually represent your data are handcrafted features. So for our domain, this, for example, means to encode the different biochemical or biophysical properties of certain amino acids in one-hot encoding vectors, for example. So you can just do some binary flag, whether an amino acid is rather hydrophobic, whether it has some sulfur in its side chain, whether it's relatively seen to the others large or small, something like this. And you can make this arbitrarily complex. You can also then introduce continuous values for some hydrophobicity scales. But all of this is highly non-trivial, because especially hydrophobicity scales are you have many of them, and this is the result of different experiments conducted in different environments at different pH values, resulting in different hydrophobic scales also. So you already see how this can already get complicated. However, the advantage of these handcrafted rules is, of course, that they are easily interpretable. So you can interpret then easily if there is a 1 at a certain position in your vector and this represents hydrophobicity, then you know, okay, this amino acid is hydrophobic. This also in turn means that only little data is required. So you rather exchange here data for knowledge. So if you understood the concept of hydrophobicity and you conducted a few experiments that gave you some direction, then you usually only need very little data for this type of encoding. The downsides of this manual feature engineering are they are definitely limited to what we know. I mean, if we do not know that something exists, we will also not measure it, and we will also not make it part of our handcrafted feature vector. So if we do not know that it exists, it will not end up in our vector. And also it is limited to what we actually can measure. So if you have a property of an amino acid that you simply cannot measure as easily as other metrics, then this will also not end up in your handcrafted feature vector. And also, last but not least, you need to be able to express it somehow in your algorithm. So this is easy if you really just consider binary states, like is a certain amino acid hydrophobic or not. But this can get arbitrarily complex. Just think about how you would like to represent, for example, proteins of different length also. This is also already not easy, that you find a fixed size representation for proteins of different length. So this can also be some task for some handcrafted features. I mean, you can just create something like the amino acid composition vector. This would be one choice. But this is a very crude way of representing protein sequences, actually. Another problem of these handcrafted features is that there is no automated way to combine these low-level features. So for computer vision, these low-level features would be an edge, for example, so just some edge detection in images, or in our case, hydrophobicity. But usually what you want to do at the end of the day is you want to find higher level concepts. So in computer vision, you, for example, want to detect whether there is a house in an image, or for us, we would like to predict protein function, for example. So you then would have to come up with some clever way to actually combine these low-level features like edges or some hydrophobicity metric for each amino acid in your protein that allows you to then predict with certain reliability the protein's function. So these downsides, and also the increasingly availability, the increasing of the available data that we have by now led to the introduction of statistical methods. So this is the second option I would say to represent your data. This is what you could call also the rise of evolutionary information. Evolutionary information meaning here that you do not only look at a single protein sequence, but that you search your query protein in today's reference databases. This can be UniProt, UniRef, or metagenomic databases, whatever. But you just search for evolutionary related protein sequences that are very likely to share the same structure and or the same function, so that you can assume that they are part of the same protein family and maybe even have a common ancestor. So this is then what we call evolutionary information, and you can condense this evolutionary information in multiple sequence alignments, where you just summarize all the related proteins you found to your query protein in these reference databases. And then you can summarize, again, the multiple sequence alignment in concepts like position-specific scoring matrices or hidden Markov model. So in the simplest case, you can just think about these vectors that you create here for each column in your multiple sequence alignment. You just count the occurrences of S at the first position, for example. Then you count the occurrences for E at this position and so on, so that you really just do some frequency analysis of how often do I observe an S at the first position in this protein, for example. So yes, PSSMs are more complex, so you also take into account pseudo counts or gap characters. But this is just one way of representing your protein sequence as some vectors by also including evolutionary information in this example. One of the problems that this method brings with it is shown here. So I just took a snapshot yesterday evening of the current size of UniProt, so one of the largest databases of protein sequences. And here you can see two things. So one thing is that we are now roughly at 210 million protein sequences in this database. And this also means that for every query protein you want to make some predictions for, or where you want to create your PSSM or hit Markov model for, you need to search also the whole reference database usually. I mean, yes, you can redundancy reduce it. This shrinks down the database size to a certain extent, but the main problem, the growth, the exponential growth that you also see here, especially in the last 4 years, is the redundancy reduction will not save us from this exponential increase in data. And this will get more problematic in the next 10 years, because then these reference databases grow exponentially, and you have to search an increasing amount of protein sequences for every query you submit to some web server, for example. This is one of the problems, so the computational aspect. The other aspect is that it's also not applicable to all proteins. So you have disordered proteins that do not adopt a fixed 3D structure, but rather adopt a 3D structure if they, for example, bind to a certain interaction partner or to some other small molecule that they need to process. And they are inherently hard to align, so it's very difficult to create large and diverse multiple sequence alignments in this step. which is crucial to have a very informative position-specific scoring matrix, or HMM. So if you have here only two sequences, this vector representation will be rather uninformative and will rather be one-hot encoding. So in the most extreme case, where you have essentially just one sequence, this becomes very similar to one-hot encoding. So yes, again, pseudo counts, gaps are, okay, gaps in this case not, but pseudo counts are taken into account. But you want here maximally large and diverse set of homologous protein sequences. And this is not possible, or it is very hard for disordered proteins. So they are usually rather shallow. Most extreme are the dark proteins, so the proteins where you do not have any information about their structure or their function, and you can also not easily infer their structure or function from homologous protein sequences. So these are really the dark matter of our proteome, or of the proteomes in general. These are just the proteins you do not know anything about, and you find also no close homolog in our today's databases that might hold some annotation. So for those, these methods are not readily applicable, or it's more complicated to apply them. So just a short summary of this second way of representing our data beyond the handcrafted features, the statistical methods now. Again, an advantage is that they are easy to interpret. So we can easily understand why a certain alignment method, some with Moderman alignment, had a certain score. So for this, we know the formulas, we know the math behind this. For substitution matrices like Blossom, we can also interpret the numbers in Blossom to understand that certain substitutions between two amino acids are just less likely than others, resulting in a different score in these backtracing matrices. So this is also, this was the point I actually want to get to. So you also can find new representations by doing this. So Blossom was actually created by some very, one of the first statistical analysis of the protein sequences that were available back then. So they aligned very similar blocks of protein sequences, and they checked for the substitutions in those. Okay, I should stop reading the chat while. Okay, but these methods also come with some downsides. So the statistical methods are still limited by what we know and what we can. Oh yeah, we're good.
Just a point. I'm going to chat to you alone if I want your attention. Everything else is supposed to sort of help others.
Yeah. I just leave it also open, then it doesn't blink. That was my main.
Yeah. I'm not sure I can make one blink only to you. Anyway.
All good. Okay, so the statistical methods, they still require us to understand the problem. They still require us to find some alignment algorithm, for example, to align two sequences, or they need us to understand that there is a concept like evolutionary related proteins, that we can summarize related proteins in families, that we can deduce from these very similar alignments also some likelihood for seeing or observing a substitution in time. So, but this is, again, we need to know it first. We need to understand it first, and then we can also try to come up with some algorithm that captures it. These things usually then also require alignment parameters. So they require parameters in general, and for alignments, this is especially crucial because this will be the foundation. The redundancy reduction of your training data set, if you now think about machine learning, will be the most crucial step of your machine learning pipeline. So you can make your supervised model arbitrarily complex. This will not buy you anything if you messed up the redundancy reduction in the first step. So to have a proper split between your train, test, and validation set, so that you really watch out that you do not contaminate your test set with anything that is similar to your training set. Sorry, Burkhard? You just unmuted, that's why. Okay. And another downside to this is, of course, you need a sufficient sample size. So, for example, the first HSSP curves were based on something around 200 proteins. So this is still a lot less than what we have available today, but you can imagine that you cannot build up these statistical methods on only 10 protein sequences. So you need a sufficient data set size at the very beginning to actually find some significant correlations in your data. If you want to come up with some statistical analysis methods. And yes, they can become computationally very expensive. As shown in the previous slides, these reference databases keep growing exponentially. You need to search for every query protein, all those proteins in the reference databases, and this is getting more and more expensive in the future. So what other options are there besides designing handcrafted features and applying statistical methods? A third option was introduced only like 10 years ago, I would say. It's called representation learning. So instead of trying to build our existing knowledge of the world into some algorithms and into some feature vectors, we actually let the machine learn itself. Representations it finds useful from the data. This is the point. So we really let the machine learn features from the data itself. And one of the closest examples, or the closest examples to our field, also comes from natural language processing, where we actually learn word representations. So for natural language processing, they also want to process sentences. They want to, for example, classify whether an email is spam or no spam, whether a Facebook post is hate speech, containing hate speech or not. You want to translate from language A to language B. So those are all problems the NLP folks encounter. And for all of them, they also need to make sentences machine readable. So they need to make a machine understand a certain English sentence in order to translate it to French then afterwards. And in the simplest, most simple flavor, Word2Vec introduced essentially two concepts. One concept called SIBO, shown here, and the other called skipgram. They are very similar to each other. One just turns the other upside down. So for SIBO, you always try to predict the center word in a window of words from its context. So you look at the two words that appeared before the center word, you look at the two words appearing after the center word, and from those you project to some hidden layer representation, and from those you try to predict the center word. Vice versa, skipgram just takes as an input the center word, projects it to some hidden layer representation, and from this hidden layer representation, it tries to reconstruct the two words that are most likely to appear before this word or after this word. So you try to predict the context from the center word. And this is a very naive approach, you might think, and why does this actually work? But you have strong correlations between certain sentence structures, between certain words appearing very frequently next to each other, and this can be picked up by the model. So, for example, New York will always appear together. It will always and your network can then learn a certain hidden layer representation for New York. But the whole point is we do not want to predict surrounding words. We do not want to predict the center word, for example. But what we actually want to have is this hidden layer vector representation that the network learned in order to do well in the task of predicting the center word, for example. So this is the main goal. We can just essentially throw away this final classification layer because this is what we are not interested in. Same goes here. We can throw away the output essentially, but all that we want to have is the vector representation learned in here for a given word. So you feed in a word and then this gets projected to a vector, and this is what we're actually interested in. But what is the goal of this? So we can do then, if we have a vector representation, we can essentially do math on words. So this is one of the very famous examples from natural language processing. So you take the vector representation of the word king, you take the vector representation of the word man, and the vector representation of the word woman. You subtract man from king, you add woman, and then you check for this new vector representation what is the word that is closest to this new vector representation? And they found actually that queen is closest to this vector representation. And I mean, this is just a very simple example to show what you can do with this now. So essentially, you can now really do math with words. And I mean, this is this example does not really, it is rather exemplary to show the power of these approaches and the information in these vectors, that they actually really captured something about the concept of genders, for example. Okay, just a short overview. The advantages of representation learning are you can leverage large unlabeled data sets. And I think this is the most important aspect. And this is also one of the reasons why it only came up recently, because now we have a lot more unlabeled data available, and we also have a lot of compute to process this, these large unlabeled data sets. Why unlabeled? I mean, you see here, essentially you do not need a label like this word is a noun or this amino acid is in an alpha helix, for example. All you need is just a sentence in the right order, or for us, an amino acid sequence, a protein sequence in the right order. And then you just feed in the two words before, the two words after the word, and then you predict the center word. But you immediately see for this, you do not need labels. You just need the sequential ordering of your data. And this is also where the most important signal actually comes from, the sequential ordering and the statistical tendency of seeing reoccurring patterns in your data. Yeah.
I do want to throw in something. I'm sorry, it's very philosophical. But the point why we need, or we use databases, unlabeled databases today and not 20 years ago, is not because they were too small 20 years ago. They were actually, so 20 years ago, the protein sequence databases already were the largest. And the problem was always to get at annotations. The point, I believe, is just a radical shift in philosophy. So whenever you talk to people who worked on natural language processing in the past, they thought about grammar, about somehow modeling rules, about somehow understanding. And to just get away with all of that, and overnight essentially to stand up and say, well, we're going to let the data learn it all. That's a very bold statement, and that bold statement is just something that happened over the last five to something like that years. Not necessarily because of the availability of the data that existed before, because of some boldness and because of some attempt to make some sense of all the data that is out there.
Yeah, yeah. To actually make use of it. I mean, now that it's there. So thanks. And yes, and I think some aspect that also might have kept people actually from doing this in the past is here one of the downsides that I list also to learn these representations. They are extremely hard to interpret. I mean, what you then get as a representation is you have this hidden layer projection in here. This can be a 512 dimensional vector, and you have no clue what the second position actually encodes for, what the fifth position encodes for. So this is just some distributed vector representation that the model learned in order to do well in this task of reconstructing the center word given the context. And yes, I mean, by doing this often enough and training this on Wikipedia, for example, or in our case on UniRef 50, I mean, you can easily map this to proteins by just thinking about the previous two tokens and the next two tokens as amino acids in a protein sequence, and then you try to predict the center amino acids. But what you always will get is here then, let's say 512 dimensional vector, this is arbitrary, you can make this whatever you feel like, could be 128, could be 1024, but you will have some vector representation. And it is not clear what position codes for what. So it is extremely hard to interpret. And I understand that rule-based systems like they were used in natural language processing before are also extremely appealing, because there you just have some if statement essentially, which says if I see amazing in a sentence, then this is probably a positive review. If I see garbage and disgusting in a sentence, then this is probably a bad review. So this is easy to interpret. These new vectors are hard to interpret. This is something, I mean, you cannot just look at it and immediately see, okay, I have these 512 numbers here in a row, and this is then an alpha helix. So this is not not possible anymore. This is less true for computer vision than it is actually for computational biology. For computer vision, you can always interpret what these convolutional kernels learned. I mean, representation learning this exists in all domains, let it be computer vision, natural language processing, or computational biology. And for computer vision, it is still something you can look at something. This is still easier to interpret, also not perfectly, but it is easier than for us, for example. So again, just spotting the right digit for inferring that a certain residue is an alpha helix within 512 numbers is not something we as humans can easily do. Another aspect is that pre-training is actually computationally extremely expensive. So you want to parse large unlabeled data sets, but then you also have to bring the compute to do so. This is even more true for recent transformer models, which I will also mention later on. So they have a quadratic dependency on memory. So this makes them extremely computationally expensive, and they also train extremely slow. And another problem of representation learning is actually, and this is getting more and more important as more of these tools actually tend to be used outside any labs. I mean, as long as they were just some prototypes, proof of concepts in labs, this was maybe not as bad of a problem as it is now, when suddenly the police in the US actually uses computer vision and automated face recognition to say whether someone is likely to be a criminal or not. Because all these models, they pick up the bias in today's databases that you use to train them. And this can be racism if the underlying database that they used for training was mostly dominated by black males, then your model is a lot more likely to predict that a black male, no matter what he did, is a criminal. Same goes for natural language processing. You train these models on large corpuses that you just scrape from the internet, and the internet is just full of trolls, unfortunately, and there is no way currently to filter these large unlabeled data sets in such a way that you really remove everything that you would consider as something that you do not want your model to actually learn. Because if you use some of the largest transformer models that are currently available, that are also used in your smartphones for speech recognition, for example. You can also use them for text generation. So you just give this model 5 words and it will produce some sentence from it, some storyline. And you really get the worst you can imagine if you just put in there something about Jews or something about black women. And this also led to an increasing debate over the last year. I think even one of the lead developers at Google, who was responsible for the ethics behind their products, quit forcefully. Let's put it this way. So they also have problems there. So this is something we always have to watch out for when we train something. For us in computational biology, this might be less of a problem because we rather have to struggle with a bias of a different nature. So we have model organisms that heavily dominate our databases. But it is still something we have to take into consideration.
Was there a question?
I actually do have a question, but I don't think I unmuted myself yet. But
Sorry?
I have a question. Can I just go for it?
Yeah.
Can you give any insight on how to actually assess these kind of biases? Because I see your point that in computational biology it's not as important as in other areas, but still, like maybe because the samples are all from male mice, for example, the data might be a lot different than if female mice were included or stuff like that.
Yeah.
Because I can hardly imagine how you could assess these biases if you don't really know about where your samples come from. Is there any way on getting that information out of predictions, for example?
So I am not aware of this bias now towards male or female mice, but I understand that this was also probably rather an example. I mean, I know that our databases are heavily dominated by bacterial proteins and also by eukaryotic proteins. But for example, archaeal proteins from archaea are extremely underrepresented. I mean, what you could do is that you set up your test set in such a way that it actually has equal amount of these three, and then you just check whether you have some differential performance between those three sets. So if you want to do secondary structure prediction, you just create a test set with 10 bacterial proteins, 10 eukaryotic proteins, 10 archaea proteins, and then you just see whether better make the test set larger so that you have also some statistical significance behind this. But I think the direction would be, at least for me, to maybe just see whether this is actually a problem. I mean, for other things like the bias in PDB towards proteins that might be relevant for biotechnology, bioengineering, or human health, there is little we can do. I mean, we can do redundancy reduction. This is already a crucial step, and this will also be part of my main message on one of the last slides. The sequence redundancy, we have very large families, very large, very similar families. And if you just train one of these devices on unfiltered data, it will also be biased towards these large families. One thing you can do is just redundancy, you reduce your training corpus at the very beginning. So do not use UniProt as you can download it, but use UniProt redundancy reduced at 50% sequence similarity, for example.
I want to sort of make the same point as Michael. I want to sort of phrase it differently. Awareness is the only thing I believe you can learn, just like in regular life. Being very, very careful and try to test as much as you can. So the organism bias you can simply try out by seeing whether other organisms are predicted by those methods. And in fact, Michael will not talk about that, but he also found out that these language models, the protein language models, are extremely sensitive to bias, and extremely, so even tiny differences between amino acid composition in all the proteins, they will zoom into slightly less or slightly more used amino acids and represent them much better. So this is something that you have to be aware of and test for your particular problem. The bias that he talked about in the PDB is a major thing. So once we developed a method where we almost stumbled over that predicting protein-protein interactions, we essentially stumbled over the fact that the system learned to predict that two proteins are from the PDB and not that they interact. That was exactly this sort of bias, and that bias came even before the world of deep learning, the world of that Michael is talking about now. So it is going to get more tricky, just like we all overloaded in our life. We have to learn to be more aware of ourselves. In science, we have to become more aware of the limits of these systems. Isabel, I like your point about the male mice, but there is this little but, and that is the remarkable reality when we look at the space of variance. If we look at the subset of all the proteins that women and men share in people and in mice, there is no way to distinguish them on a molecular level. Okay. Again, there's X and Y chromosomes, so there are some proteins that are absolutely different between men and women. But when we look at the subset of the others, there is remarkable similarity. I'm not saying that your point is not a relevant one. I just wanted to directly get into the male and mouse, which does not mean that you're not right, and which does not mean that for the next secondary structure prediction, I don't believe there will be a difference in the performance for male mouse and the female mouse. Actually, to be simple on this one here, most predictions of most proteins in the PDB are bacterial. So whatever bias we see is a bias toward bacteria. And most bacteria are fairly neutral, gender neutral. But anyway, sorry, thank you very much for your point. I rest my case.
Yannick also had a remark, right? So I just saw in the chat, yeah?
Yes, I do actually know a bit about the mouse bias because I'm doing a practicum in a lab where they analyze this data. And the problem here is actually because they want to analyze differentially expressed genes, and they want to keep it as clean as possible. So they use mice from the exact same, like the mice they use are all brothers, and they are all either one gender. So so you don't have any fluctuations in the, so you don't have any differently expressed genes. And as far as I understood it, in male mice, these genes do not change over time as much. So you have easier to analyze data. And you need to use less mice to get a better average.
Yeah, I know these inbred mice, right? But this still leaves the difference between male and female mice, right? I mean, no, Burker is shaking his head. I mean, you still have the difference in gender, right?
But again, so if I want to see whether in the molecular system there's a difference between mouse and female, yes, there is, absolutely. Right? I can clearly point my finger at certain things that absolutely differ between male and female mice. But when I look at the set of roughly 20,000 proteins that mice have, and then I ask, is there any difference in terms of structure prediction for between male and mouse, in the variance between male and female mice, then I challenge there is not. So we looked at it from the level of variance, so single amino acid variance, and we saw no difference. This is again, I'm only saying that, Isabel, because I was shocked. So my first statement was, that's the least thing you should see, right? We somehow are. Anyway, I'm not trying to put this into a philosophical observation. I always felt more like a woman, and you see that I have long hair and blah, blah. This is not where I'm going with this. I just wanted to directly respond to this. It's actually remarkable on the molecular level, on the level of the protein structures. But again, we're talking about mostly bacteria here anyway. Sorry.
Yeah, I kind of have to apologize because I think it was a bad example to go
No,
in the direction of gender here, because as you say, like the organism bias is probably much larger, or like...
No, but wait. I did not mean to say that this is a bad... I thanked you for it, and I really meant it. It's great that you made that point. And in fact, it's the elephant in the room. And I believe for you as students, this is what you have to learn. The most important lesson is to, when you see something that strikes you as the elephant, the biggest thing that we are overlooking, name it. So what you did was absolutely right. And I'm so ambitious or so patient about this, because I was very surprised when I found out. I couldn't believe it. Okay, this is why I'm so patient about it here. No. This is an elephant. It's a very good point. It's a very good example. Thanks.
Yeah. I mean, if you were to translate it to some databases that store actually mutants, and you end up with, so this is OMIM, for example, just the database that summarizes variants in human that can be related to some diseases, and then you see that maybe 80% of the proteins in there are actually only relevant for male. I mean, yes, this is now very extreme. Then this could actually become a problem. And I mean, I would not discard any bias without checking it. So you never know what these models actually pick up. So as Burkhard said, I mean, this can be something totally unimaginable, like, is this protein part of PDB or not? So this is really, the bias cannot be that far off that you should not at least consider it.
And since we are talking about OMIM, going another step into a completely different direction, the reality is that the dosage of almost all medication that you take, almost all the drugs, is completely biased by what is right for men, not for women. And that has to do historically with the way tests are designed for approval, for drug approval studies. However you look at it. But this is an example where we have a very, very, very strong bias that matters a lot.
Yeah. Same goes for children now. I mean, you see it with the vaccinations. You cannot simply assume that children are very small humans and you just reduce the dosage a bit and this will still work. So it's not that simple. Okay, but let's continue. So thanks for the comments. Much appreciated. A lot better when this is a discussion and not just me talking to you. Okay, just a second. So hard to interpret. Yes, pre-training is expensive. Bias in database. Perfect. Yes. So let's now apply this to actually proteins. I mean, up to now the examples were mostly from natural language processing. So now let's apply the story of language models, so LMs, language models, to proteins. So we interpreted now protein sequences as the language of life. So what we dubbed our paper then also published 2 years ago was the modeling modeling aspects of the language of life, or modeling the language of life in short. Just a very short recap. We are working now with sequential data. Sequential data needs specific neural networks that can handle this type of data. And one example for this is actually the long short-term memory networks, so LSTMs in short. In the figure above, you see that this is a feed-forward or a neural network, which is then essentially unrolled in time. So the output it produces is fed in again. You have a time step here at time point t. The output it produces is fed in again, but obviously not to itself, but it's unrolled over time, how they call it in machine learning. So you have here some initial starting point, x0. You feed it to your recurrent neural network, and it produces some hidden state output from this input. But what it also does, it passes on some information that it learned in here to the next time step. And at this time step, it actually takes a different input. It becomes now it gets the input token x1 as an input, but additionally at this second step, it now has also already information from the previous token that got passed over. And now the hidden state at time point h1 is essentially a mix of the new token it reads and the previous tokens. And important here, the network A is always the same. This does not change. This is always the same network which processes these inputs. But it just takes a different input at each time step, and it also gets different information depending on the time step you're currently standing at. So at time step x2, the information that is passed over here already summarizes information from the token x0 and x1. And from this, you then produce some new hidden state output, h2. And this can be essentially rolled out to an arbitrary length. So this xt can be anything. I mean, this then only has a limit in a sense that these networks can also not remember things infinitely. They will also have to overwrite their memory. So this is essentially the memory that is passed between these cells, the memory of what the network already saw in the past. And this also has a capacity limit. So usually this is limited to very few hundred time steps. So 200 residues or 200 words or tokens can usually process okay-ish, but after that, the network struggles to remember what it has already seen in the past. So this is just the very simple recurrent neural network where you really just feed in a token, you spit out some hidden layer representation, and you pass also information to the next state. A little bit more complicated version is then the LSTM. You see in here there are different modules, different weights. You have here something like the forget gate, the update gate, and the main point here is that you have two paths essentially through the LSTM network. One goes up here and receives only minimal modifications. So you see here, you only have one operation that modifies the previous information here. You have another operation that modifies the past information here. This makes the information flow from previous time steps to the next time steps extremely smooth. So you do not alter it much by adding a new token, but you let the network choose what it would like to alter about the previous things that it already learned based on a new token. This makes the flow of information in the forward pass when you process the information easier, but it also, and this is maybe even more important, makes the backward flow of the optimization. So during the actual optimization, the learning part of the neural network, you need to backpropagate the information, the lost information. For the case of LSTMs, this is called backpropagation through time. And the problem when you do not provide such an easy and smooth way to process information, you receive exploding or vanishing gradients. So there are so many operations happening in here that the gradients that you try to backpropagate through these cells over time just get infinitesimally large or small and carry no information anymore. So this carrier belt up here allows the flow of information during the forward pass smoothly and also during the backward pass. This allows the LSTM to remember longer range dependencies. But now let's map this idea to actually protein sequences. So the first step during the general representation learning is pre-training. This is pre-training on large unlabeled sequence data. This is important here. We do not need labels. Again, this is really the crucial part here. We can operate on whole UniProt. We are not limited to any bias in PDB, for example. So this is also one aspect. I mean,
yeah, I just want to jump in with this word label. So label means annotation. So label is just one of those words from the NLP field. Label essentially means that you know anything other than the sequence. For instance, its structure, for instance, its function, for instance, some of it binding or what. That's a label.
Exactly. Yes, so you really just need the plain protein sequence. So just a FASTA file, essentially. That's it. You do not need any annotations, no secondary structure, nothing. And this allows you to process also whole UniProt instead of being limited to PDB, for example. So yes, UniProt is also still biased to a certain extent, but most likely less biased than PDB, for example. So this is already a good thing. We can now move from these highly biased labeled sets to the little bit less biased unlabeled sets. But how does this now actually work? So you have here an input sequence of length L. So you have here each amino acid or each word in the sense of Elmo is fed to the neural network. On a very high level perspective, Elmo really just moves these two-layer bidirectional LSTMs over the sequence, and it always tries to predict the next token given all previous tokens. That's all that it's trained on. It really always just tries to remember what it already saw in the past, and from what it saw in the past, it then tries to predict the next word. And it does this in a bidirectional fashion, so it does not only operate from left to right, but it also operates from right to left, but the concept stays the same. It really just remembers what it already saw in the past, so this can be then, in the case of the backward pass, the end of the protein, and then it tries to predict the start of the protein. So this is essentially what it does. So in NLP, they really trained this on words and sentences, and we just translated this to our field by interpreting single amino acids in a protein sequence as words. So they are our tokens, which we now feed here as an atomic unit into our network. And our protein sequences are our sentences. So all that we try to do is, at the example of S here, for example, so S is not machine readable. So what happens here in the first layer is really just that the letter S is projected to some vector representation that is also machine readable. And then you already start moving your LSTM over the sequence. In this case, at this time step, the LSTM only knows the S. It gets projected to vector representation here so that the S is somehow machine readable. It is fed to the first LSTM layer, and the hidden state of the first LSTM layer is passed to the second. And what it then tries to predict at this position is the next amino acid, in this case, E. Same happens for the E. If you feed in the E, it gets projected to some vector representation, is fed to the first LSTM layer. Difference now is you already have information of the previous time step. So here you already have information about the S available, not only in the first LSTM layer, but also in the second LSTM layer. What do you try to predict now here? When you're standing at the E, you try to predict the next token, which is the Q. But at the time where you predict Q, you already have information about the S and the E. And you can imagine how this just continues for proteins up to an arbitrary length. So this can be a protein of length 512. So you just move the LSTM over the sequence. And at each residue, you just try to predict the next residue in the protein, given all previous residues. And that's then the pre-training step. After the pre-training, which, yeah.
I have one small question. Do you happen to know how bio-LSTMs compared to this unidirectional approach?
Sorry?
If you have a bio-LSTM, where you look at like the stuff before and afterwards, would that in theory be better or worse? Do you know that?
This is a good point. So yes, thanks for bringing this up. I think this might have be part of what I tell you later, but I think it's even better to mention it now already. I just dropped here the backward pass, so the bidirectional direction from reverse, so from the end of the sequence to the start for simplicity. So in fact, you have here a bidirectional LSTM. So this is why also here I write forward. So this is just the hidden layer representation of the forward pass. But yes, this is a good point. And in fact, this is not as simple as you might think as it is. I mean, usually you have a protein sequence, you throw some bidirectional LSTM at it, it processes the sequence from both directions, it joins the hidden states of both directions at a certain time point and predicts then helix, for example. So this is done in secondary structure frequently now. The problem is when you think about our task, we move over the sequence and we try to predict the next token given the previous tokens. If you do this in a bidirectional fashion, you already give the network essentially the labels that you try to predict. So the backward pass already saw the token that the forward pass would try to predict. So this would allow the network to cheat. So this would be super simple and would render the optimization task trivial for the network, because the forward pass would always just copy the last token of the backward pass, and the backward pass would always just copy the last token of the forward pass, and then it would get a loss of 0, essentially. But the network would not learn anything. So in Elmo, this was actually handled in a slightly different way. In order to be able to also capture bidirectional context in some way, they actually trained the forward pass separately and the backward pass separately. They only joined them by summing their losses. So this was the only time point at which the two passes, the backward and forward, actually had information about the other. So this avoided that they could copy tokens from the other. But this is, it took me a while. So this sounds a lot simpler than it is in fact, I think. So this story about forward and backward pass in Elmo is highly non-trivial. So thanks for bringing it up. But yes, so what we are actually interested in then, so let's keep it here simple. So let's only look at the forward pass. We only move our LSTM from left to right. But what we are actually interested in is not the target prediction that you see here. So we are not interested in predicting now E when we see an S. This is boring. This is not an interesting task, and this also doesn't help us when we want to predict secondary structure, for example. What we actually want to have is the hidden state that is passed here between the LSTM layers, or that is also passed here of the last LSTM layer to the classification layer, because in fact here you do not immediately go from the LSTM layer to the target, but there is some classification layer in between. So what you want to get at the end of the day is the hidden layer representation that you extract from these LSTMs, because this hidden layer representation essentially holds history of the past of the protein, because in order to do well in this task, in order to do well in predicting the next token given all previous tokens, the network had to learn to summarize all previous tokens to a certain degree at least. And exactly this information that the model was forced to learn in order to do well in this task is now what we are interested in. So we really rip off the last classification layer here. We throw it away. We do not need it usually. And what we do is we just extract the hidden state activations here and here for S, for example, and use them to predict secondary structure. And this way for E, we extract the hidden state here and here, and use this for prediction tasks. So this is depicted here. So we have our protein. I just used here again the 3D figure because it's nice, but in fact it's really sequence. So it's 1D, not 3D. Again, this is important because we can utilize the unlabeled data. For 3D, we would be limited to PDB again, which would be bad. So we really only feed in protein sequences of length L. We feed them through our pre-trained LSTM network, because this step was repeated very often. This step was repeated for all of UniRef 50. This was 30 million sequences back then when we trained it. And we kept training this over, I think it was over Christmas. So this was trained for 3 weeks or so. So you train for 3 weeks on a medium-sized GPU cluster. So from today's perspective, medium-sized. Back then, this was gigantic. It was like 8 GPUs or something like this connected. They were working for 3 weeks in a row for us, just to train on 30 million protein sequences, always just trying to predict the next token, given all previous tokens. This is the pre-training stage. So once this is finished, we rip off this last classification layer, and we are ready for feature extraction. So this is what we then do. We rip off this last classification layer, and for a given protein sequence, we feed it through our pre-trained ELMO network and extract the hidden states. These hidden states are just simple vectors. In the following, I will also call them embeddings from time to time. So embedding is just a vector representation of an amino acid, essentially. And these vector representations can then be fed into some feed-forward neural network or some other supervised network in order to produce some output, some task that you are interested in. Okay, so but now let's apply this to the prediction of secondary structure. Just a short recap, a really short recap. So we are interested in predicting secondary structure in 3 or 8 states. 3 states is really just alpha helix, beta sheet, and other. If you go to 8 states, then you differentiate between different flavors of helices depending on the turns they make. So pi helices, for example, I think make their turns in 3 steps instead of 4 for the alpha helix. And this is just a very simple sketch. This can get arbitrarily complex, as shown here on the right. So this is a PDB structure I took yesterday from a glycoprotein, actually from a virus glycoprotein. It is not the SARS-CoV-2 glycoprotein because it was already experimentally determined 4 years ago.
But spike protein from a coronavirus from porcupine.
But it's not the SARS-CoV-2, right?
No, it's porcupine.
I tried to get close, but I really forgot yesterday.
But it is a spike.
Perfect. So it was close enough. Okay. Yeah, but the main point here is really is not about. I just thought this is somehow currently the theme that is why I use this. But I just looked for something that looks complex, and for me this really looked complex. I mean, here in red you see all the alpha helices, in yellow you see all these beta sheets. So nature really came up with some incredibly complex machinery at the molecular level there. This is what we try to predict now. For every single amino acid in a protein sequence, we try to predict whether the given amino acid is either in helix state or in sheet state or in this loopy state, other, essentially, because it's just not a fixed, rigid secondary structure. So now we can use normal feed-forward neural networks for predicting secondary structure. So in this example, we have a very simple feed-forward neural network. This is the input layer, hidden layer 1, 2, and here we produce some output. In this example, we look at residue I, so at the center here at the E, and from the E, we then try to predict the secondary structure. And in this example, this would be a helix prediction. And then we move this FNN over the sequence in a sliding window fashion. And then we are at the W, for example, so I plus 2. We have a window size of 3 in this example, and here we predict a C, just as an example. What general problem would you see with such an approach? Is there anything that comes up your mind also in the direction of is a feed-forward neural network the right architecture choice for this problem? Or what problems would you see with choosing an FNN?
One that looks like this can only represent local interactions.
Why?
No, because it's an embedding, so yeah, it has the context.
No, let's move away from embeddings. Don't worry about this. Let's just think about one-hot encoding. This is a very general thing that I'm now referring to. This has nothing to do with embeddings. And you're right. So this was the right direction, Leo, don't worry. Just think about one-hot encoding, and then you have this window of size 3, which moves over the sequence. So what would be the alternative, let's put it this way, if you were now to deploy a different machine learning device, CNN, for example? Yeah, okay, I mean, Leo, you already spoiled it mostly. This is, I mean, yes, it's kind of trivial. It's only local information, yes. So with this very simple example, you have a window size of 3. So when you make the prediction at the position I, it is not influenced at all, for example, by the W down here or the E. So the prediction for position I is not at all influenced by the residue here at I+2. And you immediately see how for a very small window size like 3, this gets problematic, especially for a secondary structure prediction, which is highly local also. So you can already get pretty high performance by just considering a very local window of 7 or 11 amino acids. Especially alpha helices, for example, are defined in terms of 4. If you have a window of size 3, you will never actually capture a full turn in the alpha helix, making it really hard to actually learn alpha helices. So the bottom line here is really you throw in a certain fixed window size. So for a feed-forward neural network, you always have to predefine a certain window size, in this case 3, and then no matter what you do, you cannot allow the network to actually process information from the W or the E. So this will always be limited to whatever window size you chose at the very beginning. So now let's think about CNNs, convolutional neural networks. What would be the difference if we were to use them? So a convolutional neural network is, in its simplest form, really just this very simple convolutional filter shown here that is trained. So the numbers in here are actually what we optimize instead of the weights of a feed-forward neural network. We really here optimize the weights in this convolutional kernel or convolutional filter. And how is this done? So we have here a convolutional filter given, some input, and this is the output that it produces. And the convolutional filter is really just moved step by step over this input sequence. So in the first step, you would multiply the 0 here with the 1 down here, the 1 in this input with the kernel of 0 here, and the 2 in the input with the -1 here. You multiply all those and you sum them, shown here, and then you get as a result the -2. Then you move the kernel by 1 step. You multiply the 1 here with the 1 here, the 2 here with the 0 here, the -1 here with the -1 here. You multiply those, you sum them again, and you get a 2 out of this. And this you can repeat arbitrarily often. So suddenly you are not limited in any way to any window size anymore. The input can change. All that you define is the convolutional filter, and this is moved over the sequence. Now you could argue, okay, so this is very similar to actually defining here this feed-forward neural network and then just moving it along the sequence. So yes, in the first step, this is in fact really very similar. This only becomes different once you stack multiple layers. Because here in the example, we have here an input protein sequence running along this axis. And you have here a convolutional kernel with window size 3. So it takes SEQ, for example, in the first step and produces a 1 from it. Then it moves 1 step down. So it takes the E, the Q, and the W, and it produces the -1 in here. And this is where it already gets, this is the main difference actually between feed-forward neural networks and FNNs in my eyes, because the feed-forward neural network would just take SEQ as an input, and it would run all the way through to the output. It would already produce some prediction like helix at this point. So it would always be limited to the SEQ. It would not see any other information. Now the main difference to convolutions is instead of producing immediately an output at this point, it first runs along this axis, along the sequence axis, and produces for each element in here also hidden state activation, feature map, no matter how you want to call it. Essentially, it just produces some new learned representation inside the network. It first processes the whole sequence, creates one new representation, and then once you stack another layer, this layer can already operate on the whole new sequence representation. And here the point about stacking multiple layers comes into play, because at the first layer, you only have SEQ available as context information that you can learn from. SEQ produces the 1. Then you move down 1 step. EQW produces -1. But when you now think about this kernel absorbs the 1 at this position and the -1 at this position. And as a result, it can already access information ranging from the S to the W, meaning that it can actually access information within a window of size 4. So you increased, by adding one more layer, you increase the window size by 1 also. Just by stacking 2 of these kernels, both have size 3. So you did not change the size of the kernel, you did not change the window size or anything, but by just stacking them. And then now you can imagine, so this kernel absorbs essentially all these inputs, and the 0 at this position would already hold information about this E. So if you have this kernel and it takes the 1 here, the -1 here, and the 0 here, you see how it can already operate on this context. So it already sees SEQWE in the next step when it produces its output. And this continues. So this is just, I mean, the more layers you stack, the larger the perceptive field will get. Did this get clear? Just saw Matthias worry. Okay, then I might have misinterpreted your reaction. Yeah, Yannick?
I have a question. So are you predicting like the single instruction now for E or for Q or for S?
Yeah, this is now, okay, perfect. So you essentially already, I might, for the future, I might introduce yet another slide in between those. Now this is a little bit overloaded information-wise. So one concept I wanted to show you here is how you get more and more information, or the more layers you stack, the larger your receptive field will grow. But actually what you're right, I mean, in fact, if I were to predict secondary structure, I would optimally want to have a secondary structure prediction for each of these elements in here. So if I have a sequence of length 8, in this case, I want to have also an output of length 8. So this should align. I want to have for every single residue a secondary structure prediction. So this is exactly the problem of the method shown here in this very simple example. So now that you know this, Yannick, any idea on how to avoid this?
No,
for everyone, I mean, not necessarily only Yannick. Everyone can answer this. I mean, the problem is also not inherently limited to CNNs. This is also a general problem of sliding window approaches.
I mean, I guess the start and the end are not impacted as much because you're only there once or twice.
Yeah, but I mean, I still want to have a prediction for S, no matter whether it's now less important, more important.
I mean, it's just taken into account less often. That's what I wanted to say.
Sorry, I didn't hear. So Matthias, you also mentioned something, or Linus?
Yeah, I was just. Sorry, you go first.
I just wanted to say with the sliding window approach that I thought maybe if you slide from the first position to the last position, the middle positions you basically take into account often in your window, but the first and the last positions, depending on your window size, if it's a bigger window size, then the first 5 positions, for example, only well, they matter less than the ones in the middle. That's what I want to say.
Yeah, but I mean, do you really meet them less? I mean, you also see them once here, and then you move on once, then sees these ones once. So it's really less about how often it sees these things. It's really rather about having an alignment between the input and the output. So you want to have an output for each single amino acid in the input. And currently what happens here is with every single layer, we lose also essentially one residue position. Matthias, you also had a comment.
Yeah, I mean, I was gonna say that you could just add some padding, maybe.
Sorry?
That you could just add some padding in order to avoid the loss of dimensionality.
Exactly. So this is where I wanted to get at. I mean, you can just artificially add zeros here at the beginning and at the end. And this avoids then also the loss of the reduction of your input size. So this is the problem. This would also get this would get worse over the layers you stack. So with every layer you stack, you would lose essentially half of your window size. So there is a formula. I mean, here you see for window size of 3, you always lose one position at the start and one at the end. You lose here one at the start, one at the end, one at the start, one at the end. And this keeps going essentially until you either have here an empty vector or you produce some output. This is totally fine if you have here, for example, an image and you just want to classify at the end whether this image contained a cat or not. But for us, this is different. For us, this is more like a sequence translation task. We want to translate from a string or a sequence of amino acids to a string or a sequence of secondary structure elements. So if we have an input of length L, we also want to have an output of length L. So we really have to explicitly force the network to avoid this reduction. And as Matthias already mentioned, the common way to do this actually is to add artificial zeros at the start and at the end. So for a window size of 3, you only add 1 0 at the start, 1 at the end. And then the first residue here takes actually the 0 into account. And then you produce already a 1 for the S. So you do not lose any residues at the start or at the end. If you had a window size of 5, you would also need to add 2 zeros up here and 2 at the bottom. If you had a window size of 7, you would have to add 3 up here and 3 down here. So depending on your window size, you need to choose the appropriate padding so that you still get an output of length L for an input of length L. And yes, this is, I think, did this get clear now, or is it still confusing? Leo?
I think it's quite clear, but I have a question. Sorry, Leo, you wanted to go first?
No, it's not. It's a question that's not so clearly close related. You go first.
I think mine is not so close related too, but
yeah, then just one of you start. I mean, this is I also answer unrelated questions.
You're using the term kernel and convolutional filter. Is that actually the same thing?
Yeah. Oh, thanks. Okay. Yes. This is really the worst thing. I mean, names. This is just also residue. I think residue is something maybe is more common amongst you than it is amongst computer scientists, but we just use it on a regular basis. For us, so residue is just amino acid in a protein. We take this for granted. But if I then talk to them and I tell them I make a residue level prediction, this doesn't tell them anything. Same goes for convolutional filter or kernel. Same.
Is your kernel always the same? Because you have one on the slide, but is it always the same size and actually the same elements for each layer that you stack?
So it is the size that I define at the very beginning. So I can make this also 7 or 5 long or 9. This is something I can define at the very beginning. So this is a hyperparameter that I choose. And the values in here, they get actually, I mean, they are just integers here for simplicity. In fact, they are continuous values. They are randomly initialized and they get optimized. So this is exactly the weight essentially if you translate it to normal neural networks, this is the weight you would try to optimize and learn during the training process. So this is not the same for all layers. It will get different.
Yeah, thanks.
Hopefully, at least. I mean, if your network learned the same thing with 2 kernels, this would be kind of bad. This would be a waste of resources. So, but it could happen. I mean, if you train a very large network on very little data, it could end up learning similar things. Okay. So this was one question, but we also had another one, right?
Yeah. So if you're using this kind of padding,
Yeah.
does the prediction for the beginning or the end of the proteins actually get worse because you're using this using less information about the context?
This is a good point. So short and honest answer, I did not check explicitly. I never checked whether if I only look at the first 5 residues or the first 3 or the last 3, whether they have a different performance than the ones in the middle. What I saw, and this is rather a gut feeling from looking at the predictions, it did not heavily affect the performance. Usually those residues are anyway unresolved in PDB structures, so they have these wobbly ends because they chop proteins into smaller, easier to digest units that are easier to capture with their X-ray experiments or their cryo-EM experiments. And this way, when you create these chopped proteins, you end up with some ends that are relatively flexible. So usually you do not have a really good ground truth information on those anyway. I understand that this answer might have rather raised more questions than it answered. But the bottom line is, from what I know also from machine learning, it does not heavily affect the performance. So the network actually learns to ignore this information because it's all zeros. So you can imagine what you would do at the end of the day is just think about it this way. You flip around this convolutional filter and you have here now 1, 0, -1, and then you just move it down here. No matter what value is here in this filter, if you multiply it by 0, it will give you 0. So this has no effect on the final performance, and also the loss that gets back propagated is essentially 0. So the network learns by itself that this is irrelevant information. But I also saw already concepts where they explicitly added here a binary vector with 1s and 0s, where here would be a 1 for the S because this is a valid amino acid, and a 0 up here because this is a padded element. I did not try whether this actually has any effect on secondary structure performance. This is something
actually most likely for longer for shorter proteins it will have an effect, and this is a very good point. We should try that. Because in the old-fashioned padding a way of doing padding that made a big difference.
Okay. Yeah, I mean, I just know from, again, looking at these predictions by just visually, I did not see there a heavy imbalance, but I understand that this is an extremely weak metric of assessing a bias. So rather make it quantitative. Oh, okay. We have here a feature that's raising the hands. That's cool. Yes.
I would have another question. I don't know if you already mentioned that, but is the operations in the kernel or convolutional filter, are they restricted to group operations, or like with the 0 padding, we do have this like neutral element appending there, but it plays a little bit into the point of how does it affect the beginning and the end. But does it restrict as well how we can build our kernel, or?
Yeah, I mean, you can imagine if your average sequence length is 20 and you create a kernel with size 100, then you will pad a lot, unnecessarily a lot. But besides this, I would not know how this, I mean, just make it a reasonable, make the window size a reasonable fraction of the sequences you actually want to operate on, as well as the features you want to detect. For secondary structure, I mean, you can look at the, I want to include this plot actually. You have there a distribution of average helix length and average sheet length, for example. I mean, if your task is now to predict secondary structure and you see that 90% of your helices are actually shorter than 8 residues, if you count consecutive length of helix stretches on your sequence, then it might be sufficient to just have a kernel size of 9 or maybe 11 to be a little bit above it. But I would not see a reason to make it then 150, for example. So I would rather design these kernels based on the task you want to predict. So a colleague of mine who will, I think, give a lecture in a week or two, Konstantin, he is doing structure prediction, and there the most important information is actually to predict long-range contacts. So contacts that are extremely far apart on the sequence, but are very close in 3D space once the protein folds. And when you want to capture those, you definitely need a much larger window size than what I needed here for secondary structure. So I really used a window size of 9, but Konstantin used much more. Yeah. Okay, so I think I need to move on now, sorry. I hope this answered your question, Marcus. Okay, perfect.
Thank you.
Gerne. So just let's shortly recap advantages or disadvantages. Disadvantages of FNNs and advantages of CNNs. So for FNNs, the receptive field is limited to the chosen window size, and deeper layers will definitely, by definition, have absolutely no access to any information beyond this window. So in this example, SEQW, the output will always only see SEQ. It will never have any chance to see the W. And for convolutions, you can design the window size accordingly. I mean, yes, you can also adjust here the input size, but the more CNN layers you stack, the larger your receptive field gets. And the advantage is that this is computationally extremely efficient because you only have to stack these very small filters of size 3, for example, and if you stack 10 of them, you would already have a extremely or a relatively large receptive field. While if you were using here input of size 10, then this would already get resource-wise a lot more intensive. So you create a lot more free parameters here because you have these all against all connections. So you really have much more free parameters if you wanted to capture a similar window size with an FNN. And yes, so CNNs can operate on sequences of arbitrary length, so it doesn't matter whether this is now only 8 tokens or 100 tokens, it will just move over the whole sequence until it sees the end. That's it. So this is extremely flexible also for capturing sequences of variable input length. So now just as a short overview, here again we have our protein sequence, 1D information, not 3D information. We feed it through our pre-trained Elmo language model. So we encode the S, the E, and the Q first in the first step as these vectors so that it's machine readable. Then we move the LSTMs over them. And what we then extract are the hidden states from the LSTM states. So this is then what we call our embeddings. So this is just a vector representation for each amino acid. So for each amino acid, you would then have not an S at this position, but a 1024-dimensional vector. Same goes for the E. You also have a 1024-dimensional vector for the E, but with slightly different numbers and so on. And this can be readily be used as an input for your CNN. To predict, for example, secondary structure. And again, here you then need to add the zero padding to have an alignment between the input length L and also the output length L. So we applied this concept for secondary structure. Here you see along the y-axis the 3-state accuracy for predicting alpha helices, beta sheets, or other. And here we have 3 test sets: CASP12, TS115, and CB513. Here you see a bunch of methods that reach something like state-of-the-art performance. They all use evolutionary information. They all use the explicit database search I introduced at the very beginning. So they always search the query protein against some large reference database. They compute these multiple sequence alignments. This summarizes the protein family information. You squeeze this into some PSSM or HMM, and this is then fed to a relatively complex supervised network. In the case of NetSurf P2, this is a stack of CNN layers and LSTM layers, and as far as I remember, they really had CNN layers with larger window size than 100. So this is really, they put it to the extreme here. And this is currently reaching something that could be considered state of the art with 85% on these two test sets. And our method with Elmo, with the LSTMs, is somewhere here. So this was 2 years ago. This was kind of nice. We were operating on single protein sequences, so this was cool. We still got some reasonable performance. We got close to JPROT4, for example. We also outperformed the predecessor of NetSurf P2, NetSurf P1. And we didn't have this bottleneck of the database search, but our performance was significantly worse than state of the art. So this is definitely not what you want to have. So how is this actually then, or how did others improve over LSTM-based approaches? And this is what you could call the rise of transformers. So this is a different model, but why would you now want a different model? LSTMs work perfectly with sequential data. They have this inductive bias of processing sequential information. So this is perfect. The problem of LSTMs is they have to move sequentially over the sequence in order to produce some hidden state for a given token that can be passed to the next step. So you can immediately imagine how this is a nightmare for parallelization. This is not possible to parallelize. You really have to, in fact, absorb one token at a time, feed it through your LSTM, produce some hidden state, and this hidden state can then be produced and fed as an input to the next time step. So this gets super slow for longer sequences. So if you have a protein of length of 1,000, then this already gets super slow. The problem is also that long range dependencies need to be remembered. Contact prediction is one of the projects we are also working in our lab with, and there you need to capture long range dependencies with hundreds of residues of sequence separation potentially, and you need to remember those, and the LSTM would have to remember it to actually be able to predict also then or to detect some constraints between these. Also, as mentioned previously, there is no real bidirectionality during pre-training. I mean, if you have two LSTM layers, one is the forward pass, the other is the backward pass, you cannot immediately concatenate the hidden states because otherwise the neural network could cheat. So the forward pass could just copy tokens from the backward pass and vice versa. And transformers were introduced to solve these issues. So they allow parallel processing of the whole sequence. So you really feed in 1,000 tokens, and in one feed-forward pass, you get the embeddings for 1,000 tokens. So you need not to sequentially unroll the LSTM for 1,000 time steps. This is really one pass, all parallelized, if extremely efficient, but also extremely memory intensive. This allows also to model long-range dependencies because internally you perform all against all comparisons. So each single residue in a protein is compared to all other residues in this protein. This is amazing on the one hand because no matter how far apart the residues are on the sequence, you will still compare them and potentially detect some correlation between them. Problem is, you can imagine this squares quadratically in memory. So in order to do this all against all comparison, you essentially need to create an L by L matrix internally. And you do not only create one of those guys, you create actually 768 of these all against all comparisons in our biggest model. And the big advantage also is they really capture bidirectional context during pre-training. So not like LSTMs where we only summed the loss. The forward pass actually only had information to the tokens before it, but not after it, vice versa for the backward pass. Now transformers can really capture the whole bidirectional context. And how do they do this? So let's just look at it from a very bird's eye view, high level perspective. So similar to Elmo, if you just see it as a black box, similar to Elmo, BERT also acts as a model that allows you to represent your data. So for an input sequence, an input protein sequence, it just spits out some vectors that are readily machine readable. So this is the very high level picture. And the main difference is, instead of LSTMs, BERT uses a stack of transformer encoders, so some attention layers. I will not go into details now due to time reasons, but in a nutshell, they really perform these expensive all-against-all comparisons. Performance-wise, great. Memory-wise, horrible. So this is a kind of a trade-off where you need to find a sweet spot in between. But what is actually BERT's training objective? Because this is also different. Instead of predicting the next token given all previous tokens, like Elmo, BERT introduced something that's called masked language modeling. And there the task then becomes to reconstruct corrupted input tokens given all other tokens. So in practice, this worked such that for a given input protein sequence of length L, 15% of the tokens were just randomly replaced by some special token. And this corrupted sequence is then fed to the transformer model. And the objective is then to reconstruct the corrupted tokens from the non-corrupted context. So, I mean, 85% of your sequence stay the same. So I am now already over time, I just learned. If you need to run, then run. I will really try to keep it super short now. So this is just a very high level overview. You give some input sequence, you project it to vectors, you feed it to the transformer model. The transformer model gives you some embeddings. So these vector representations, in our case, again, 1024 dimensional vectors, these can be fed to a supervised network like a CNN, and then you can already predict secondary structure. So here there's also a way to actually create per protein embeddings, but this is not important now. What you can also do with these, yeah, this is also not important. I think this is the main bottom line, actually, because we also got some new results a few weeks ago, actually. So this is pretty new. It also gives you some kind of a historic overview how this developed over the last 4 years, roughly. So again, we have here 2 different test sets, CASP12 here, a new test set that was strictly non-redundant to the training set that I created over here. We have 2 times NetSurf P2 performance on these 2 test sets. NetSurf P2 is again this method that uses explicit database search. And then we have here ProtVec. This is uses the uncontextualized word2vec. This means that it will always produce the same embedding for a given input word. So for apple, it will always produce the same vector representation, irrespective of whether you mean the fruit or the smartphone. Same goes for amino acids. Word2vec always produces the same embedding for the same amino acid. We then introduced context, essentially, by training the NLP language model Elmo, which is LSTM based on UniRef 50. This created different embeddings for the word apple depending on the context. And here you see this nice jump in performance from 63 to 74. And the next step was then to replace LSTMs by transformers. This was done by ProtBERT. Here you also see again a decent jump in performance, even though not that much given the additional compute we threw at it. And then what we published just a few weeks ago are these new T5 models. So T5 XL with 3 billion free parameters. So these are really gigantic models. They were trained on more data. They had more free parameters than ProtBERT and also slightly different transformer architecture, which then here is 77% or over here 82% when trained on these large metagenomic databases like BFD. And in the last step, we then fine-tuned the T5XL BFD version, so the one that was trained on completely highly redundant data on UniRef 50. This is what I meant earlier when I said one of the most important biases for us is actually redundancy in databases. So we have extremely large, very similar protein families, and you do not add any, or for most tasks, you do not add much information by just showing the machine learning device protein sequences that are 99% sequence similar. And this is as true for these language models as it is for any other machine learning device. So the largest jump actually in recent time that we observed was from here, from the T5 version that was trained on metagenomic, highly redundant metagenomic data, to the non-redundant version of it trained on UniRef 50. So now we are here at 81%. The state-of-the-art is somewhere at 82. For the new set, we are at 85. State-of-the-art is at 84. So by now, we can essentially predict secondary structure from single protein sequences without searching these databases at the same performance level as it was previously done by this explicit database search. And here are just some more detailed analyses. It's not that important. Last slide now, don't worry, you can soon leave. The point here is we plot here the Q3, the free-state accuracy against the number of training steps that we trained these models on. And you see here this nice correlation, and we did not observe yet that this actually plateaus or levels out completely. So yes, it gets more and more expensive. I mean, here is a logarithm, so this is a logarithmic correlation. So this is getting increasingly more expensive to squeeze out the last few percents. But it also seemed like we did not reach a ceiling yet for what these representations can actually learn. Our current ceiling was rather the grant that ran out, so we did not have had any compute anymore. But this could potentially be improved by just training longer. And of course, also using different transformer architectures. So now there are already so many more. So this is the last slide. Thanks for your attention and sorry for running over time. Thanks for the participation, especially. So this was a lot more interactive. I enjoyed this. And yeah, don't touch this one. So this is no Pokemon, even though it looks a little bit like this. Some of the students who already participated in the winter semester lecture might remember this one. So this is the blue dragon. It's highly poisonous, so don't touch. And that's it. Thanks.